Good morning, everybody, and welcome to this Microbiology Society President's Roadshow. For anyone who doesn't know me, my name is Peter Cotgreve, and I have the best job in the world because I am the Chief Executive of the Microbiology Society, and it's my real pleasure to welcome everyone this morning to this roadshow. This is the first President's Roadshow of 2023 and the first to be held for an international audience following the success of those that we held both in person and virtually over the last 2 years around the UK and Ireland. And this roadshow is focused on West Africa. It's not a part of the world that I'm very familiar with, so I'm really excited to hear the contributions from a variety of microbiologists from Benin, from Nigeria, from Burkina Faso, from Ghana, and UK-based scientists with strong links to West Africa. And this event is an opportunity to find out about the Microbiology Society and the opportunities available, and we're going to hear some real experiences of microbiologists who've taken advantage of some of those opportunities, and we're going to listen to talks from local researchers in West Africa, and I'm really looking forward to that. So please do make the most of the opportunities today. An important part of the roadshow is the opportunity to network, and there will be the speed networking session later in the day, and do please take advantage of that to make connections with other people who are here at the roadshow today. My job now is to hand over to the chairs for the day, who are Philippe Sessou and Victorien Dugnon and Omololu Fagunwa. And I'm now going to hand over to Philippe, who is going to chair the first session.
Thank you, Professor Peter. As you know, my name is Philippe Sessou. I am associate professor at the University of Microbiology in Benin, West Africa, and I am chairing this event with Professor Victorien and Dr. Omololu from Nigeria. So I'll give the floor to Dr. Professor Victorien to present himself also. Dr.
Thank you very much, Professor Sessou. I'm Victorien Douillon. I'm an associate professor in microbiology at the University of Abomey-Calavi. And I'm heading a research lab. I'm also the head of the International Cooperation Department at the University of Abomey-Calavi. Thank you very much.
Okay, the floor is to Dr. Omololu from Nigeria to present himself also.
Thank you much. Good morning, everybody. My name is Omololu Fagunwa. I am a research fellow at Queen's University, Belfast in UK. I was formerly ECM committee member within Microbiology Society. I look forward for today. I believe we have a very successful event. Thank you.
Okay, thank you, Dr. Omololu. So we will start the proceedings with the talks from the Society's President, Professor Gordil Bezra. This will be followed by talks from some of our members, where you will find out about the great benefits of the Society membership and publishing with us. There will be a speed networking session in breakout rooms after the Society's talks, where you will be able to talk to speakers about getting more involved in Society's activities. And after speed networking, we will hear free scientific talk from our invited speakers. So important housekeeping, we will be recording the majority of today's, and the recording will be uploaded to the Microbiology Society website for members. Please feel free to turn to turn your camera on, but ensure your microphone is muted unless speaking in a breakout room. This will avoid any interruption for today's speakers. Please use the chat box to introduce yourself and network with other attendees. You can also use the chat box to contact society staff should you be experiencing technical difficulties. We will be using the Q&A function for a question during some of the talks. So please direct any question to the Q&A box when available. We will only be using the raise hand function during the speed networking session. So please refrain from raising your hand during the talks. We have enabled closed captation for this event. If you need to access these, press the show captation button. If you want to hide them, you can press hide captation. So I will first give the floor to Dr. Victorien. Do you want to introduce the first talk? Thank you.
Thank you very much, Dr. Sesu. So to start with the proceedings, it's my pleasure to introduce Professor Godel Beza from the University of Birmingham. With him, we will go through the insights into his journey as a TB researcher. And I would like to show that as well as being the society's president, is also Badrick professor at the Institute of Microbiology at the Hello.
Hi, Victoria. Okay. Sorry, we were having some issues with your sound, but I think it's working again now.
Oh, is it fine now?
Yes, it's okay now. Thank you.
Okay, I'm so sorry. So I would like just before to give the floor to Prof. Besra, notice that his talk will be followed by a 10-minute question and answer just after the presentation. So please feel free to let your question in the chat, and we will come back after that. Professor Godel Besra, dear President, this is my pleasure to introduce you. You have the floor. I think we can all see your screen.
Thank you. Thank you for the kind introduction. So
flow to. I would just like to stress that there will be no question and answer session for Prof. Lucas talks, but you can ask your question to her during the speed networking session. Professor Iruka, this is my pleasure to give you the floor. Thank you very much.
Thank you very much, Victorian, and thank you to the organizers for inviting me. So I have the pleasure and honor to be a senior editor for one of the Society's journals called Microbiology. This is one of the reasons why I've been invited to share what the Society is doing in terms of publishing for the community. So as many of us know this, the Microbiology Society has several journals. The very first was set up in 1947, and that was Microbiology. There's also the Journal of General Virology, the Journal of Medical Microbiology, International Journal of Systematic and Evolutionary Microbiology, Microbial and most recently Access Microbiology. So these are really high quality journals. They are either open access or hybrid, as you can see on the slide. Hybrid means that in some cases, authors can pay an author fee and then have their article be open access. So a lot of the material that's available in the Microbiology Society journals is actually free to read. And this is excellent for those of us working at institutions where getting subscriptions to subscription-only journals is a challenge. And in terms of wanting to read them as scientists, obviously we want to read the work in our field, but also we want to use a lot of these resources for education of our students. For example, Journal of Medical Microbiology has these JMM profiles that I use a lot in my classes that overview a particular species. And so it's nice to have a lot of this resource that's open. And also, the society is a not-for-profit society. So even when resources are needed to publish, they're needed so that they can make material available to the same community. So for over 75 years, I mean, microbiology, which started in 1947 and a year ago became fully open access, is the first journal. And so the society has a long history of publishing good microbiology science for the community. The articles are high quality, they're peer reviewed, and the society is developing and expanding and strengthening networks so that members can actually share their knowledge. I encourage you, if you're a scientist that is publishing your work, to consider the society journals, and also to consider reviewing for the journals when invited by editors. So there is a move within the society to transition to sustainable open access so that anyone, anywhere can access the latest research, and not just be able to access it, but also so that authors can publish in this open access mode. So some of the recent open access milestones that you might be interested in is the relaunch of Access Microbiology, which is a great open research platform. And one of the things that you might be interested in is that Access Microbiology welcomes a range of science, including negative results. So if you have negative results that came from robust and rigorous science, this may be a place to send your article. And by the way, if you do submit to one of the society journals and your article is of good quality, but not necessarily good fit for that particular journals, editors will typically figure out whether this is a better fit for a different journal and ask you whether your paper can be referred to another journal that the society publishes. So as I mentioned earlier, microbiology is now fully open access. And there are a number of models which I'll go into, including the publish and read model, that give access to authors who might want to publish open access, but might not necessarily have author fees in hand. So what do I mean? The author support for open access in the journals. First of all, if your institution has a publish and read agreement with the Microbiology Society, that means that every author affiliated with that institution can publish open access for free. There are not a lot of African institutions that are doing this yet, but there certainly should be. And there's certainly institutions around the world that are taking advantage of this. So if your library is able to set up such agreements, you could actually reach out to them and ask them to consider applying to Microbiology Society to arrange a publish and read agreement with your institution. The other thing is that there are member discounts on article processing charges. So whenever you find yourself faced with an article processing charge that you can't pay, you should certainly be asking whether there's a discount that fits for you. And I think what's most relevant to those of us who are authors from the African continent is that there are fee waivers for authors who are in countries covered by the Hinari program, which many of us will be familiar with. This includes Group A countries, which you can see is most of the countries in Africa who have free access to publish open access their accepted articles in society journals, as well as Group B that receives significant discounts to be able to publish open access in the journals. And if for any reason, any of the examples I've run through doesn't quite fit you, and you think you need assistance for open access, it's always important to reach out to the journal. There may be something that you're not aware of, or that the society can offer in terms of a fee waiver. So I'm really hoping that this will encourage colleagues to publish in Microbiology Society journals. We have quite a few articles authored by African authors, but not as many in terms of the representation in science. So I'd really like you to consider this for your next journal. And this is just a list of the countries that are eligible for Group A access, as I showed you on the map before, which gives you free access to open access publishing, as well as Group B, where you would have a significant discount, low cost access. So as mentioned by Victorian earlier, there won't be a Q&A after this talk, but I'll actually be available in the breakout room along with several staff from journals in the Microbiology Society to answer questions about the journals, including what sorts of things we accept, what the peer review process looks like, and the impact that you can get from open access, which includes expanding the reach of your science so that other people can read your work without difficulty, increasing impact of the work that you publish, and also supporting the community, because everything that the Microbiology Society does is really for the promotion of the science of microbiology. It is a non-profit entity. So I want to thank you for your attention. Thank you for the opportunity to be able to share what the journals of the Society have to offer today, and happy to take any questions during the breakout session. Thank you very much.
Thank you very much, Mara. Thank you, Professor. It was a real pleasure to hear from you and the journals of the Microbiology Society. We are really grateful. Thank you again. So I think we will move in our trip, and I'm very pleased to introduce the second society talk. This will be about supporting the careers of early career microbiologists. This talk will be done by Raphael Gallet from the University of Sheffield in UK. Raphael will discuss his career, the support received from the society, and the details of how to get more involved with the society as an early career microbiologist. For this talk, there will be no question and answer session, but Raphael will still be available to ask any question during the speed networking session, as for Prof. Iruka Okeke. So, Raphael, if you are ready, I'm very pleased to give you the floor. Thank you very much.
Okay, thank you so much. I hope you can hear me.
Perfectly.
Like you said, I'll be talking on supporting the careers of early career microbiologists, ECM. And I'm a PhD student here at the University of Sheffield. So yeah. Is my slide moving? I don't know why my.
No, now it's not moving. Maybe you can close it and open again. It will work.
Okay.
You share again.
Okay. Let me try and share it again.
Yeah. Good. Is that? Okay, now we can launch it.
Okay, it's now moving, yeah?
Not yet. Okay, now it's moving.
Okay, so that was a brilliant talk by Professor Bisra in Kiruka. And here I just want to tell you brief about myself. So I have a BSc microbiology from Nasarawa State University, Kefi, Nigeria. And then I did my master's in pharmaceutical microbiology from Ahmadu Bello University, Zaria. I moved to the UK for my PhD, PhD in microbiology and immunology, which is resident in the School of Clinical Dentistry here in the University of Sheffield. And then I joined the Microbiology Society sometime in 2019, and then became a member of Early Career Microbiology. And then I became an executive in Early Career Microbiology sometime in 2021 till now. So my journey as a microbiologist, if you can see this slide, at the undergraduate level, I was sciences where microbiology, zoology, and plant science and biotechnology are different units in biological sciences. So I was the vice president of my department sometimes in 2007 and 2008. And then in 2008 to 2009, I became the first microbiology president in Nasarawa State University, Kefi. Then Society for Microbiology as well, sometimes in 2017. And for my postgraduate level in ABU Zaria, where I did my pharmaceutical microbiology and master's level, I was the assistant class rep. And then here in UK, coordinator for oral microbiology, sometimes in 2019 to 2020. And I'm also a member of oral microbiology and immunology group here in the UK. So like I said, in 2019, I joined the society, Microbiology Society, and then I became an ECM member in 2020. And also the board member for microbiology today is actually a magazine for Microbiology Society where we give update and other stuff about Microbiology Society, and became the scientific conferences panel representative of the ECM in Microbiology Society in 2021 till present. So my engagement with Microbiology Society here in the UK, I have quite a lot of engagement, but in this slide, I just have a brief overview to give you. I was involved in Microbe Talk where I talked about myself, for example, we talked about what early career microbiologists face, especially international students that are here in the UK, the kind of challenges they can get involved in.
Sorry to interrupt. We're having some issues with your sound. It's just cutting out sometimes. So if you could just, I don't know, maybe speak a bit louder or yeah, maybe try that. Thank you.
Okay.
Sorry.
Okay. I'm sorry about that. I hope you can hear me better now.
Yes, yeah, we can hear you. Yeah, thank you.
Oh, okay. Thank you with that. So I was saying some of my engagement were involved in organizing the ECM summer conference that held in Sheffield sometime last year. I also attended some society annual conferences where I presented a poster. Here you could see this is me presenting my poster at Belfast, our last conference that we had last year. Again, some of our focus meetings and partnership. This other picture by this side is our ECM Away Day in London. You could see myself and some of our Microbiology Society staff. And the picture in the middle is my lab mate from our lab here in Sheffield that attended the ECM summer conference. And then the last picture here is our oral microbiology and immunology group meeting that we had at Birmingham sometime last year as well. So these are some of the focus meetings and partnerships that I've been involved with. And so, like I said, I'm the scientific conferences panel representative. And then Joe Kite is our chair or our chairman in the executive committee of ECM. And so for me, what I do basically is to attend scientific conferences, panel meetings, and division days, to also discuss issues that are relevant to ECM and report back to our committees or council, as well as I got involved in organizing ECM summer conferences and other related matters. So that's what I do. But in the ECM, we have different other committees that are saddled with different responsibilities. But then this is part of my own responsibility for what I do. And so the ECM Scientific Conferences panel, like I said, this is just like an overview of what I do, oversee the delivery of the scientific conferences program to support the society's 3 main objectives, as well as to work with other committees and divisions to try to see how we could discuss relevant and attractive topics that are important to early career microbiologists. So why you should join us and some of the benefits as an early career microbiologist or as a young microbiologist, because my focus in this talk is I'm focusing on the younger microbiologists that are coming up. Why you should join us is you have, like I said earlier, you have exclusive access to free places and selected training courses for core skills. You also have discounted access to ECM focused conferences and meetings, including flagship ECM summer conference. So if you're a member of ECM, if, for example, we're doing our summer conferences and you're supposed to pay to attend some of those things, you have discount that you as a member, you enjoy. And then you have eligibility for an ECM forum poster prize if presenting at annual conference. So without being a member, you won't be able to participate in this competition, this poster competition for ECM. But if you're a member, you could actually participate and if possible, win a prize. And then eligibility to apply for the ECM co-chairing scheme at the annual conference. I will tell you more about co-chairing later, but if you are a member of ECM, you could actually apply to co-chair a session in annual conference. Co-chairing will help you to learn from other seasoned microbiologists or seasoned researchers how to chair sessions in annual conferences, which is going to look good on your CVs and the rest. And then you have the right to run for a position and vote for your elected representative. So without being a member of ECM, I wouldn't have been able to become a representative of the ECM in the scientific conferences panel. So if you want to be an executive of the ECM, you have to become a member first. And without that, you won't be able to vote or be voted for. And again, you have access to our quarterly newsletter, which shares upcoming exclusive opportunities for forum members. So there are quite a lot of benefit for you if you become a member of the early career microbiologist. So these are some of the benefits here you could see. I talked about you participating in poster presentation. Here is actually my certificate where I won the prize for best poster prize at the last annual conference we had at Belfast, where my talk or my poster won as the best poster for Access Microbiology. And being a member of Microbiology Society and ECM member, again, I was able to publish in Access Microbiology. Professor Nkirika was talking about you publishing in Access Microbiology. So these are publications that ordinarily you were supposed to pay in other places. But being a member, I was able to publish my work in Access Microbiology Journal. So I had like 2 or 3 publications with Access Microbiology Journal. And again, I wrote like a blog post about the irreversible gum disease, which is periodontitis. And here, this last other screenshot by this other side is actually a letter of appreciation from Microbiology Society, acknowledging my role in co-chairing a public health session at our annual conference that held sometimes in 2021. So these are some of the benefits that you stand to gain if you become a member of early career microbiology. And again, being a member, you have opportunity to apply for grants. For example, you're in West Africa, you want to attend our international conferences, which is going to be very good for you, gives you exposure where you're going to meet with other microbiologists to network. You could actually apply for a grant, and then the maximum award is about £750. But then if you're within UK or different other places, there are different price amount that you could actually apply for. Myself, I've benefited from Microbiology Society grant twice. This is me attending a conference, courtesy Microbiology Society. So yeah. And the Microbiology Society, we have different ways of giving back. So how can we give back? First, you have to join. And to join, all you needed to do is to become a member of Microbiology Society itself. And then you go down on your dashboard, is where it will ask you if you want to become a member of ECM. Once you just click yes, that's all. You don't have any special fee that you need to pay to become a member of Early Career Microbiology. All you need to do is to indicate interest. And then you could actually publish in society journals like Professor Nkiruka said earlier. She also said if you're available and you want to become a reviewer, you could actually review for our society journals. Myself, I was involved in reviewing for Access Microbiology Journal last time. And you could actually give feedback and attend networking and feedback events just like this one that we're doing. So join us and become a member. You're going to have quite a lot of benefits. So what are you waiting for? You can join us today. If you're on LinkedIn, you can just go to LinkedIn and search for Microbiology Society Early Career Microbiologist Forum. I'll leave this for a while. Yeah, you could search on LinkedIn. You could just follow us and you see some of our updates, what we do in early career microbiologists. But most importantly, not just joining us on LinkedIn, you have to become a registered member that will actually contact you one-on-one. So thank you for listening, and I think I'll stop sharing now.
Thank you very much for this interesting talk. So now we will go for a short break of 10 minutes. It was 15, but let's meet at 12 and 15, please. So we just go for a short break. Thank you very much.
Just to confirm, that's 11:15 UK time, 12:15 West Africa time.
Benin's time.
Yes, in Benin. Yeah, thank you, Victoria.
Thank you.
We'll move on. So welcome back, everybody. We will start our second session. I hope you are enjoying these great talks. So for the next, we have a presentation. Get involved, become a champion. This presentation will be done by Dr. Joseph Apolasey from Cape Coast Teaching Hospital in Ghana. He's a microbiology society champion. So Joseph will discuss the role of champions with the society, within the society, please, how to become one, and his experience of being a champion. For this session, we have no question to be done, but Joseph's talk will remind delegates that they can ask him a question during the speed networking session. Thanks, Joseph, for this talk, and then introduce the next society talk, which we have two speakers. So Joseph, please get the floor.
Okay, thank you very much, Prof. Philippe, for the introduction. My name is Joseph Akolache. I'm here to present the role of champions and my personal experiences on becoming a champion and to urge you to Microbiology Society. This is the outline of my slide.
Oh, Joseph, we seem to have lost you.
So a little.
Oh, we can hear you
Hello.
again. Hello. Okay, we can hear you again now. Sorry, it was just cutting out there.
Sorry, my connection is a bit unstable.
Okay.
So here's a little about me. First and foremost, I'm a proud champion of the Microbiology Society. And trust me, this is one feat I always make sure added to my curriculum vitae. And I bachelor's in molecular biology and biotechnology from the Cape Coast University, which I completed in 2018. Since then, I've been employed as a research scientist at the Cape Coast Teaching Hospital. My work focuses mainly on infectious diseases and clinical microbiology relating to those of bacterial origin. Since then, I've had about 6 years experience in microbiology, specifically antibiotic resistance or antimicrobial resistance in general. So to introduce you to the champion scheme, I thought of two ways to do that. And so I wanted to take you through the first option, which I would share now. So when they told me to give this talk, I thought, okay, could probably just direct you to the website, which has now been boldened on the screen, where you go through and read what the champion scheme is, what champion, etc. And then I'm done. So I would probably hold a record for one of the shortest talks in the world, but then this won't give us the engagement we needed in terms of actually getting your attention to become a champion. So I came up with another option, which is option 2. Now, with this option, I could share my unique champions experiences and do this from a or truly benefit or truly understand what it is to be a champion, know who a champion is and what exactly we do. So we are going to explore this option a bit more. Currently, there are 107 champions worldwide, 8 from Africa and 6 from West Africa. So meaning out of all the African champions, West African champions make up about 75%, if my math is not wrong. So this is the cool graph I decided to add as a signature scientist, because we know scientists love charts and data. But I still haven't told you who a champion is. So I'm going to say from my perspective, I first joined the society with the influence of my mentors, Dr. Vicky Choka, who's with the NHS Blood and Transplant, and Dr. Brad Spiller, who's at Cardiff University. So they prompted me to join the society. And the reason was we're applying for a grant to fund some research, which was subsequently successful. But since then, I wanted to know how could I give back to the society after they had given to me. And that's when I found the champions team applied and was subsequently accepted. So for me, who a champion is someone who seeks greater involvement in the society and wants to contribute to the society. Now, as I applied, there were two qualifications I had that made me successful in applying to be a champion. First was having a fair idea about microbiology, and this was in 2020, so I wasn't as knowledgeable as I am now. And the second was enthusiasm. And for enthusiasm, this was the most important quality which led me to actually be accepted as a champion. So once you have enthusiasm, that is one great factor which can make you a champion of the society. And then now to what champions do, I'm going to give you 3 practical examples based on my personal experiences to demonstrate what we actually do and the benefits you get from being an active champion. The first was a presentation to clinicians and nurses in our hospital. So this was a paper we published with my mentors and some other researchers in collaborating institutions, where we found some environmental contaminants in inpatient ward environments, which were resistant to carbapenems like meropenem and cephalosporins, such as ceftriaxone. And we had to communicate the phenotypic and genomic methods we used to these clinicians in order to spare some initiatives or interventions to control them. So having to translate biofilm associated biofilm-associated factors and virulence factors to the level where clinicians appreciate these things was quite challenging. But with support from the microbiology champion scheme, we're able to do this. And this, in the long term, improved my science communication skills incredibly, because it's not just about a publication, it's about going back to the community where you conducted the research and being able to speak to them in terms they understand to drive change. So these are some of the pictures where we were in the wards speaking to doctors and nurses with the microbiology society flyer clearly captured in the picture. And this was me leading a part of the presentation as part of the facilitators. And for the second example, we held an event on research conduct. We're training PharmD students to conduct points prevalence surveys to assess their prescribing patterns within our institution. And in doing this, it's really improved my organizing of training events, especially for the planning and evaluation. So usually people do host events, but they don't have any stringent evaluation policies or any stringent evaluation plans. But for being a champion, you actually have guidance where they give you a form and ask you to state how you are going to evaluate it. So you tend to now quantify your achievements for each program you do organize. And this is the picture where I was taking them through the steps of conducting point prevalence surveys, which subsequently led to a successful data collection exercise with the PharmD students, solely led by them. And for the final example, I attended my first conference outside the shores of Africa last year. That was the ECMID, European Conference for Clinical Microbiology and Infectious Diseases. And it's quite lonely when you're going for your first conference and you don't have anyone to sort of be in community with. So I was lucky enough to find volunteers at the Microbiology Society stand, and that's where I spent most of the free time in between talks for the conference. So it helped me foster a sense of community and belonging. From people busily rushing from one session to another, I always had a place to go back to talk and feel at home, even though I was a million miles away from home. And these are some of the pictures, including a picture I took at the booth with one staff at the Microbiology Society. So in summary, becoming a champion through organizing events, through what we do, I benefited by improving my science communication skills, by improving my event organizing skills, and by benefiting from feeling a sense of community and belonging. And so I hope this will also encourage you to be champions as well in the society. And thank you very much for your time.
Thank you so much, Mr. Joseph, for your great presentation, where you talk to all participants how to become a champion, which kind of activity we can do as a champion. And in your conclusion, you said that when you are a champion, you can improve your sense communication. You can organize a lot of events with the support of the Microbiology Society. So I will tell to participants to become a Microbiology Society champion, because there are a lot of opportunities you can obtain by joining this group. So we will move to the next presentation, which will be done by two speakers. There are Dr. Marguerite Klein from University College Dublin in Iran, who is a grants reviewer and editor of Access Microbiology, and also sustainability committee member. She will be assisted by Dr. Adebare Adeleke from Modibo Adama University in Nigeria. They will do a presentation on making the most of your membership with your society grants. I would like to remember you that Dr. Marguerite is one of our grant reviewers, and she will talk about how to prepare well your application and what they look in what they look for in an application. There is no question to be asked, but you will have the opportunity to ask a question during the speed networking session. Please, Dr. Marguerite and Dr. Adebare, you have the floor for your presentations. Thank you so much.
Okay, good morning, everybody, and thank you very much for that introduction. So I'm going to talk today about grants that the Microbiology Society can offer you as a member, and how to make the most of your membership with the Society by applying for these grants. So there are lots of benefits of being a member of the Microbiology Society, and the grants are just one way, but they are a fantastic way to make the most of your membership. So just some idea of how applying for a grant from the Microbiology Society can actually support you, and the number of people that do avail of these supports in the Microbiology Society. For example, up to 400 people were supported by grants to attend society conferences, and then a further 160 were supported to attend other conferences that weren't run by the Microbiology Society. And attending conferences is a great way to network and make friends, but it can be expensive, and we do need support to do this. Over 20 people traveled across the world to visit collaborators. Up to 50 students were able to take summer studentships, do research projects over the summer. And then 10 people were involved, supported for local activities, and a further 10 people then for education and outreach activities. So if you want to apply to the Microbiology Society in order to make an application for grant support, there's certain things you need to look at. And I would advise you to look at the website before you apply for any grants. First of all, to see what grants are offered. But before you apply for a grant, do check, number one, that you're eligible, because different grants are targeted towards different communities within the society. So before you apply for a grant, do make sure that you are eligible. And look at what the grant covers. So what is it that you need? What is it that you're applying for? And does this particular grant scheme actually cover what you are looking for? There's an application process, and that's very important that you follow that. At this site, it will tell you exactly how you can apply for the grant. And then there's award criteria, and I'll talk a little bit more about this in further slides. But the award criteria are very important, because no matter how much how well you write your grant or what you're looking for, there are certain criteria through which your grant will be scored. And you must ensure that you address each of these criteria in order for your grant to be awarded. And then there's just some details about how these awards are administered. So all these details are available on the microbiology website. So I have reviewed a number of different grants in the Microbiology Society. I'm a member of the Sustainability Committee, and I've looked at a number of different grants. So all applications are reviewed blindly by at least 2 referees. So when I say they're reviewed blindly, that means that the person reviewing the grant does not know who has written the grant or where it has come from. Referees score the grant according to the award criteria. So that's why I said it's really important that you look at the award criteria, because the referees are going to give your grant a numerical score, and that is based on how well you have addressed each of the award criteria. So if you don't address a certain award criteria, then you're not going to get a score for that, and your score will be lowered. And they also submit comments. So, for example, if for some reason your grant was not awarded and you're not successful, you will get feedback and comments, and this will help you then when you apply for further grants, and you'll know why your grant wasn't awarded. The actual decision, though, as to who gets the grant is made by an award panel. So the referees review the grant, they give a score, and they make comments. But there's another panel that comes together then, and they discuss the comments and the scores and decide, and where there may be a discrepancy between 2 reviewers in terms of the scores and the comments. And this award panel will then review the grant themselves and look at it and decide which comments are fair and reasonable and actually reflect what's written in the grant application. And the comments from the reviewers then are sent to the applicants. And remember, some grants may be competitive. And that's why it's really important. You want to get that highest score possible, because sometimes there's a large number of applicants for a particular grant. There's a limited amount of money that can be allocated, and you have then the awards are made to the very best grants. Even though your award may be eligible and you may have addressed each of the award criteria, if the grant scheme is competitive, if somebody has made a better application than you, they will be funded. So you want your grant to be the best grant possible, as with all grants. And so here I have listed for this particular scheme, a research project grant, what the award criteria are. And again, I've taken this from the website, so it's very clear what each of the award criteria are. First of all, the project must be relevant to microbiology. So if you have, you're looking for a grant for a physics project, for example, or whatever, it's not going to be funded. But if you can outline how relevant this work is to microbiology, then you've addressed that award criteria. There must be clear scientific merit in the project, and this must be underpinned by good scientific reasoning. So there must be rationale for the work you're doing and for how you're doing it. The scientific objectives need to be clear and realistically achievable in the proposed time frame. So you might have very clear scientific objectives, but you might insert enough work that, that could take a whole year to be done. So, and the time frame for the grant scheme might be 12 weeks or something. So be realistic. Are you going to achieve the work that you've outlined in the proposed time frame? The approach you're going to take, the experimental approach, needs to be clearly described and also technically feasible. If you're going to a host laboratory, does that laboratory have the expertise and the equipment to support the successful completion of the project? So the person reviewing the grant won't know that. So it's up to you to tell that person what expertise is available in that grant, in that host lab, and what the equipment available is that you need, and demonstrate that it is there in the lab. There must be clear benefit to both your laboratory and the host laboratory. So you might outline how there's very clear benefit to you, but also outline how your own laboratory is going to be benefit. So when you go back to your own laboratory, how you're going to share your expertise and the knowledge that you've gained, and also how the host laboratory is going to benefit from having you there. And full and reasonable costings need to be given. So reasonable costings. So think about what expenses or money that you are looking for. Is this reasonable? And don't leave things out, so put in the full price, but also really important, look to show that you have costed these, that the costings are reasonable, and that value for money is demonstrated. And also on the award site, and this is really worth looking at, there will be an example of an application that was awarded. So this is really useful, and you should look at this and see how a successful grant, how each of these award criteria have been dealt with in a successful application form. So this is just an example. I took the research project grant as an example, but for each grant that is listed on the microbiology website, there will be individual award criteria for that scheme and an example of a successful application. So these are really worth looking at before you start to write the grant. So this is, so as a reviewer then, that reviewer then gets that grant application that you have submitted, and they will also get a scoring sheet with each of the award criteria outlined on that scoring sheet. And you can see here that there are certain scores that the reviewer can award to each of the criteria. So one of the criteria here that you're scored on, for example, for a travel grant, would be your current position. So if you're an early career researcher, such as postdoctoral researcher or a PhD student, you're going to get full marks. 4 marks for that. Now, you can't do anything about your current position. You either are or are not, an early career researcher. You might be a research group leader, in which case you're only going to get 2 marks. But you do have control over all of these other criteria. So benefits to research. So to get full marks, you have to provide specific details such as topics, concepts, and issues regarding the research work and its relevance to the conference event that you're attending. So make sure, you really want to make sure you get those 4 marks and not 2 marks or 1 mark. So pay really careful attention to that. Professional development, again, full marks. The applicant is specific as to how this conference or event is going to ensure that they reach their future professional goals and how it relates to your current research interests. Costs, that you provide a detailed and a complete budget, including travel and lodging, etc., and that you show that you have sought to get cost-saving measures. So if you have, booked your flight to travel to a certain event, early, and you've got a reduced airfare, or you've booked lodging, or you're sharing accommodation with somebody, highlight that you've looked for the best value and the cheapest options that are available. And your abstract then, if you're looking for a travel grant to provide to go to a conference, is this abstract directly relevant to the conference, and outline how relevant it is to the conference. So, if you tell the reviewer, then they don't have to, spend lots of time looking at the conference, looking at the abstract, and trying to decide whether it's relevant or not. You tell them how relevant it is, and then it's very easy for them to make up their mind as to the relevance of the abstract to that meeting. So make it easy for the researcher, for the reviewer, to give you full marks for each of those sections. So as a result of receiving a society grant, lots of members have, received lots of benefits. Now, for example, because I'm a member of the Microbiology Society and I'm involved in the Microbiology Society, my students have presented at Microbiology Society conferences, and they've got funding to attend those conferences. Students have come to my lab and they have done summer research work, and they have benefited enormously from that scheme. And I also have attended research conferences supported by the Microbiology Society. But there are lots of different ways that you can benefit. So if you are a member, you should make use of that membership. So look at the grant schemes and look and see, what do I want to do? Which of these grant schemes can help me? And apply as soon as possible. Okay, thank you.
Thank you so much, Dr. Marguerite Klein, for these great presentations. Very, very insightful presentation. So we'll move to the next presentation, Dr. Adebari from Nigeria. She is one of our travel grant recipients. She will have to give her talk. Thank you so much, Dr. Adebari, for your presentation. So please get the floor. Dr. Adebari.
Okay. Oh, I just unmuted myself. Good day, everyone. I hope you can hear me.
Yes, it's okay. It's okay.
Okay. Dr. Adebari Johnson Adeleke from Modibo Adama University, Yola, Nigeria, southeast of Nigeria. Actually a recipient of a travel grant award, and I'm here actually to give some testimonies, from what the previous speaker, Dr. Klein, has talked about. Yeah, I got the grants, travel grants. I was able to attend the FEMS Conference on Microbiology held in Belgrade, Serbia, and also online. So I was able to present two of my abstracts, which are displayed on the screen. The first one is ion associated electrogenicity in a clinical strain of Klebsiella pneumoniae and correlation with virulence potentials. And the second one is optimization of alkaline sugarcane bagasse pretreatment for sustainable bioethanol production by Candida tropicalis. Somebody from a place like Nigeria, where we usually don't have enough funds to attend international conferences. Thank God for, I'm happy for a society like Microbiology Society, which actually gave me the grant that made me to be able to make my presentations, communicate about our research in my laboratory to the entire world. Dr. Klein has done so much of what I want to do, but I will just try to go through some stuff now and emphasize on some grants that are available. But I hope this is moving.
It's not moving at the moment.
Slide is not moving.
No, maybe try closing it and opening it again.
I should remove. Okay, it's moving.
Oh yes, it's moving now.
Okay, so through the through my attendance at the conference, I was able to get important feedbacks from our research, which have been very, very productive for us to continue the research. And also, I was able to know some other things that people are doing in my related research interests, quite a number of innovative approaches to research. Also, me participating at the conference was able to boost my self-confidence, getting to realize that you are not so doing bad, talking with other scientists from all over the world. And also, I was able to make some international connections. Now I receive emails from scientists from Europe, and we try to share ideas together and trying to see if we can apply for a joint research grant. So if you are a resident in West Africa and you are interested in applying for grants from the society, which grants can you apply for? Well, I need to first inform you that you have to be a member of the society for you to be eligible to apply for the grants. So try as much as possible to go online and fill the form as a member and subscribe. You have so many things to benefit. One of it is the travel grant, which I benefited recently, and the maximum award, if you get the travel grant, is £750, and with it you can present your work at a scientific or educational meetings. You can attend a short course or training events, and you can, the grant can support both also virtual or online events. You don't have to go there physically alone. You can also use it to attend online conferences. That is travel grant. We also have the research visit grant, which the maximum award is £3,000. And this can allow you to undertake a visit to a research laboratory in the UK or Republic of Ireland. This is especially very important for, very relevant for PhD students or postdoc students. Maybe they are especially from this part of the world, in West Africa. If you have some aspects of your work that we don't have enough equipment in your country or your institution, having a host in the UK, Microbiology Society will be happy to sponsor you so that you can visit the foreign laboratory and do that work that you couldn't easily do in your country. The funds can support the travel, accommodation, and subsistence of a member, and also help you to buy some project consumables. We also have the society supported conference grants. The maximum award for this grant is £2,000, and you can use this to organize a conference, whether in person or virtual, in any field of microbiology. And as a member, you can organize the meeting alone or in partnership or with other societies. The funds here may be used towards funding the cost of invited speakers, travel, and accommodation, And for a virtual event, funds may be used to cover delegate fees for invited speaker. We also have the International Development Fund. The maximum award for this is £5,000. Institutions in West Africa can host a society member to give short lecture courses or laboratory training, especially maybe you just acquired a particular equipment in your laboratory and you don't have the technical know-how. A member from another location who knows how to do it can visit your lab and train the staff in your lab to make use of such equipment. It can also assist in the purchase of consumables or small pieces of equipment in a lower, middle, or low-income country where a society member works. There is also society conference grants. This particular grant is used specifically for society events. For instance, Microbiology Society will be having its annual conference very soon. This kind of a grant you can apply for it if you're a member so that you can attend the conference. The annual conference, the maximum amount for the annual conference is £380, while some other focus meetings you can apply for as high as, you can apply up to £250. Under the Society Conference Grant, we also have one that is Caring Cost Grants. And the society is so magnanimous that there shouldn't be any hindrance. Maybe you have children you are caring for, there shouldn't be any hindrance for you to attend an academic and education meetings. So you can apply for up to £500 to take care of your children or child, or maybe you have some other caring needs while you attend a particular conference. But this can only be used when you are attending the society conference, the Microbiology Society conference. So if you consider yourself as somebody who needs a grant, that will be applying for a grant from Microbiology Society soon, I would like you to note some of these points. You need to get early information on conferences and training workshops you desire to attend, because this you might need to have a long-term planning. Most of the time, they announce conference maybe like 10 months ahead of the particular dates. You need to acquaint yourself with this information so that you know the right steps to take. You also have to get yourself acquainted to available grants and deadlines. You can see all this in the website of the society. You should try and also make sure that you are up to date with your membership subscription to qualify for grant applications. For you to apply for a grant, you have to be up to date in your subscription. If your subscription has expired, you may not be able to apply for a grant. I'm aware that people like, at the moment, people from Nigeria might be having difficulty in paying subscription because of the barrier in making international transactions. One of the things I would suggest that you can do if you are from Nigeria, because presently the banks in Nigeria don't allow Nigerians to make international transactions with their local card. You can contact a friend or relative that you know abroad in UK. They can make the payment for you, then you arrange how you return it back to them in Naira. That's what I do. So you also need to dedicate considerable time to provide logical answers to the questions in the application forms. Don't just rush and answer the question. You need to answer them before you submit, read, and see if you can make it better before you finally submit. Then once you are okay with your answers, do not procrastinate. Submit your application as early as possible. So I implore you to join the society today. Take your time to explore the various career benefits the society can offer you by regularly visiting the website. You'll be glad you did. The website is displayed there, and I could see it has been displayed in the chat box. So try as much as possible to visit the website of the society to explore the various benefits that you stand to gain. Thank you for listening.
Thank you so much, Dr. Adibari, for this great presentation. So I hope the participants are enjoying this great presentation or thought. I will thank you very much for this. And also I will thank Dr. Marguerite Klein for her presentation. There is no question to be asked by the participant guests, but they have the opportunity to ask a question during this video networking session. So we will move to the next speaker, who is Dr. Michael Okuru from the Federal Polytechnic Ida, Nigeria, Building Committee member. So he has to give us a presentation focused on how to join a committee. Dr. Michael, please have the floor.
Okay, good day, everybody. I hope, can you see my slide?
Yes, it's okay.
Okay. My talk is on how to get involved and join a committee. Is it moving?
Yes.
Okay. My name is Dr. Michael Okuru. I have a PhD in food microbiology from an university in Nigeria. That is Michael Okpara University of Agriculture, Omodike. I've been a member of the Microbiology Society and also a member of the Building Communities Committee. I'm a member of the editorial board of the Microbiology Today and a member of the Champion Scheme Review Working Group. So I'm a champion. Now, let's talk about how I got involved in joining these committees. Number one was quite my interest in the strategic objectives of this society. We get to see those strategic objectives and how I was happy with them. Now, how does this work? I used to have regular emails from all the committees, groups that I've joined, and then vacancies and election information always communicated through mails. And with regular visit to the society website, one gets to know clearly what exactly I was looking for. And you can see the society website displayed there. So over there are a lot of details, just like previous speakers have said. So I used to attend meetings regularly. I receive relevant materials from meetings via my email. So in all, I've never found any barrier. So I want to let anybody who is interested know that if you want to join these committees, no logistical barriers to participation. Of course, there are a lot of opportunities when you get involved, many opportunities if you are members of committees, panels, or divisions. You'll be meeting others, and as you do that, it's a kind of networking as well. And of course, you know that your net worth is equal to your network. Then there's access to grant, the last speaker just mentioned. And of course, there's increased efficiency and influence. You will influence the society in very many ways. Effective collaboration, and I mentioned network earlier. And while you collaborate, there's a lot of knowledge sharing, having to develop yourself professionally, and then you have leadership development, which leads to long-term sustainability and resilience. Of course, here you also find equality, diversity, and inclusion, always practice in every aspect of what we do. So the society is so diverse, and then it helps in many career developments. Committee members are supported to attend events like annual conferences and even other meetings. Now, I mentioned earlier that my first interest was on the strategic objective. And let's look at the strategic objective one. Now, summarize them. Strategic Objective 1 has to do with understanding diversity of members, strengthening existing relationships, gain access to new communities, unlocking potentials for international collaboration and global knowledge sharing. That's Objective 1. Now, let's look at Strategic Objective 2 that has to do with harnessing local knowledge for worldwide implementation, advance understanding of microbiology, and then champion contributions by microbiology where you address global challenges. Now, Strategic Objective 3 talks about recognizing global differences. Of course, you build on existing financial and governance funds, sustainability, and resistance through diversity income streams, as well as increasing efficiency, robust mechanism, monitoring and evaluation. Of course, when you look at all these strategic objectives, this is exactly what makes a life worth living. And for making this life worth living, all you have to do is to join the committee. Now, what committees are available for you to join? There is this Early Career Microbiologist Forum Executive Committee, and then it supports the career and professional development of microbiologists. Among us today, you find so many early career scientists, microbiologists, and of course, many here today have testified to what it means to be in the Early Career Microbiologist Forum Executive Committee. Now, Building Communities Committee oversees the delivery of Strategic Objective 1, as I mentioned earlier. So when you look at everything that I mentioned in Strategic Objective 1, the Building Communities Committee is meant to deliver this strategic objective. Then we have the Impact and Influence Committee that oversees the delivery of Strategic Objective 2. And then the Sustainability Committee oversees the delivery of Strategic Objective 3. These are beautifully organized for career development, for networking, and whatever one really needs for development. Now, apart from these committees, we have panels and divisions. These panels oversee specific societies' activities. Now, you have the Scientific Conferences panel. They oversee the content of all our scientific conferences. And then you also have the publishing panel. These are made up of editors-in-chief of all the journals. They come together to make up that panel. And then somebody has shown us, one of the speakers has shown us the need to publish or participate in our journals today. Then the members panel, there are quite a lot of underrepresented communities who are expected to be members of this society. Now, this panel is responsible for them. Let me say the first committee, that is the Scientific Conference panel and the Publishing panel, they report to the Building Communities Committee. Now, there are also divisions that look at specific activities of the society. You have the eukaryotic, the Irish prokaryotic, and virus. These are various divisions, 4 of them in all. Now, these divisions are there. We have specific people with various professions in each of these divisions. And then they also represent regional participation to be effective in this society. Now, looking at all these divisions and groups and committees, one, you can participate in any of these. Now, it is important that you expand your reach, you increase your impact. And how do you do it? You can only do it when you join this committee. And to join the committee, I want you to watch out the nominations process for vacancies starting in January for 2024. It will open in March 2023, which is just next month. So if you watch out next month, you see a lot of openings where you can apply and become a member of one of the committees, divisions, or panels for this society. So this is how we join the society and get involved in all our activities. Being a member of the society is a comprehensive lifestyle of its own. So join this society today. Thank you for listening.
Thank you very much, Dr. Michael, for your great presentation. And during that, you discussed about the various opportunities available to join committees, divisions, and panel at the Microbiology Society. Again, thank you so much for your great presentation. So now we will move to the speed networking. But before that, I would like to thank all our speakers for their great presentations. So now we'll have the time to discuss about the talks again. So for this speed network session, which will take place via Zoom breakout rooms, all the speakers from the society focus talk will be available to answer questions of you participants with the support of staff from the Microbiology Society. So if you would like to ask a question while in a breakout room, please use the raise hands function and staff will invite you to speak in turn. So the speed networking session is for only 30 minutes in length. And if you would like to visit all the breakouts rooms, you have about 5 minutes to visit each room. Society staff will broadcast a message every 5 minutes to give you the option to move to the next room. But feel free to stay in the same room for longer if you wish. If you go into a very busy room, please also feel free to try another that is less busy to ensure you can ask any question you have. So when the breakout rooms open, you will be given the option to join a breakout room, and you can choose from the following 5 rooms. The first room is for publishing for the community. The second one is for early career microbiologists. The third one is for champions camp. The fourth is for society grants. And the fifth is to join a community. So select the room you want to visit and then click the blue join button. If you are having difficulty to join a room, please stay in this room and select the raise hand, and a member of the society staff will assist you. So when the speed of networking is coming to end, you'll have to say what you want. Thank you so much. Thank you for your attention. Mrs. Fifian? Hello?
Oh, yes. So the breakout rooms are now open. So feel free to jump into the rooms that you want to go to. Charles will be here to help if anybody needs assistance moving into a room, and I will be in the champion scheme room if anyone needs me there. Okay.
Thank you so much. So the speed networking is okay.
Yes.
You can go on. Yeah.
Yes, do go ahead. Yes, thank you.
I hope break was very nice. So now we are going to the next step of our journey to welcome our first scientific talk and keynote speaker. It's a lady, Dr. Esther Deguenon from the University of Abomey-Calavi, who will share with us the experiences of the research unit in applied microbiology and pharmacology of natural substances in conducting antibiotic resistance studies using One Health approach. So Dr. Deguenon is an invited speaker. She will have 25 minutes presentation, and we will follow up with some question and answer. So feel free to leave your question in the chat. Dr. Esther, if you are ready, you can be sharing your presentation, your screen. Okay.
Thanks, Dr. Duyon.
You have the floor.
Thank you. So I'm Esther Deguenon. I'm PhD. I'm affiliated to the research unit in applied microbiology and pharmacology of natural substances. I'm laureate of the L'Oréal UNESCO Prize for Young Talent, Sub-Saharan Africa, and also co-coordinator of a project funded by the World Academy of Science and the Islamic Bank Development. So it's my pleasure to present you action we are taking in the unit to combat EMR in Benin and West Africa. So we will follow a plan that we talk about the research unit. We will make a review of the One Health approach in the fight against AMR. And I will present some research on AMR using one approach in the unit. We will also show the limitation that we have to conduct AMR research. And we will also talk about the involvement of the research unit in fighting AMR in the three sectors of One Health. And then we will present the prospects in the fight against AMR that we have in the unit. So to start, let's talk about UMAFA. Let me present the unit. So we have been created in 2017, and we are a trans-faculty research unit. We are in the research laboratory in applied biology in Polytechnic School of Abomey-Calavi in the University of Abomey-Calavi. And then we are physically and administratively located on the campus of the University of Abomey-Calavi. Now, let's talk about our vision, that is to be a national unit of excellence where scientists work together to find solutions to health and environmental problems of importance in Benin and in Africa and around the world. Our mission, most importantly, is to conduct cutting-edge basic biomedical and public health problems relevant to the needs of population, really relevant to the needs of populations. So we have two major research programs. One is the fight against antimicrobial resistance that have many components, isolation of pathogenic microbes from different sources, phenotypic and molecular characterization, detection of resistant and virulent genes, and application of bioinformatics tools to biology. And also, we used to promote medicinal plant use. And for this, we used to conduct ethnopharmacological survey, chemical characterization of medical plants, biological and toxicological characterization, and development of phytomedicine. But also we use for the sustainability. of medicinal plants in Benin and West Africa. So because we used to work with plants, and then we have to be eco-friendly. That is what is really important to us. Now let me show you where we came from and where we are now. The building A was where we started in 2017 in a pre-made building, and now we are in the building B. We have a lot of sessions inside: molecular biology, standard microbiology, and phages session, and some of other discipline. So let's make a review of the One Health approach in the fight against AMR. So everything has started in 1990, where awareness of stakeholder of the broad articulation of health issue made starts a lot of things to lead to the One Health approach. And in 2004, we had the One World, One Health integrated approach by the Wildlife Conservation Society. And then in 2005, creation of the European Center for Disease Prevention and Control. One Health initiative has started with interdisciplinarity in animal, human, and environmental health. So in 2008, we had the adoption of the One World, One Medicine, One Health concept, a conceptual and strategic framework to reduce the risk of infectious disease at animal, human, and at the level also of the ecosystem. It's important to highlight that 60% of the 1,400 human pathogen comes from animal origin, and 75 of them are emerging diseases that are zoonotics. And also now in 2009, we get the formalization of the ecosystem dimension of the One Health Eco-Health Association. And then we have the tripartite note between the FAO, the WHO, and the OIE. So the vision is a world capable of detecting, containing, eliminating, and responding to risk to animals and public health from zoonotic disease or animal disease that would compromise food sufficiency using collaborative, cross-disciplinary approach and strong partnership. And this has been adopted by numerous countries. For the One Health, we have 4 core professions: veterinarians, doctors, pharmacists, and ecologists. We have an historical focus on zoonotic infection disease, but animal disease are able to jeopardizing the sector, and we have a lot of things that can be implemented using this historical focus. So the major issue now is the resistance of bacteria to antibiotic, of parasite to antiparasitics, and all through human and animal share the same pharmacopoeia. So toxicology plus ecotoxicology will give environmental health, and we have also pathology associated with exposure to chemicals. In the One Health approach, we can have a lot of cause and transmission route of antibiotic resistance bacteria. And we can have at human level, we have a hospital that is the main point of dissemination of antibiotic resistance. And we can have contact between patients, with visitors, nurse, all the staff of the hospital. We can have resistant bacteria that appear after antibiotic treatment, and then can be transmitted from animals, animals, and vice versa. Also, we have the tourism that can be a vehicle of dissemination of antibiotic resistant bacteria. And most important, we have the transmission between animals and human, between human environment and between environment human and animals. So we have a lot of zoonotic disease that are not really controlled in our framework. Now we are going to talk about the specific research on AMR using One Health approach in our research unit. So we have a lot of studies that we go through and that help us to have some result that can be used as preliminary for more big research. So we have the study one who talked about the prevalence and antimicrobial resistance of Campylobacter coli and Campylobacter jejuni, isolated from pig guts, pig feces, and surface swab for the cutting table of slaughterhouse and tavern in southern Benin. It's important to know that all these studies has been done through classical bacteriology and molecular biology using classic PCR. So for this research, we made the relation between pigs, the animals, the slaughterhouse, and the restaurant. So the relation between animal, environment, and people eating this kind of meat. Also, we had a second study who made us know the implication of bats in the transmission of zoonotic strain of Salmonella in Benin. This research was really special because we found a lot of Salmonella strain that has been carried by the bats and that are frequently transmitted to human because of the proximity of this kind of bats, this kind of animals and human. So we also use the classical bacteriology and PCR methods. And a third study had method assess of the bacterial pollution and detection of antibiotic-resistant genes in Benin. And this was the case of the Lake Nokwe. So as you can see, all our work are between human health, animal health, and environment. And for this study especially, we found a lot of contamination of fishery products. And the issue is that in Benin, a lot of people used to consume this kind of product coming from lake and other kind of water courses that we have. So the method used was also the classical bacteriology and PCR. For the fourth study, we assess the presence of resistant genes detected from the environment in some selected food produced in Benin. And we find a lot of bacteria, pathogenic bacteria, and overall, they were resistant to numerous antibiotics that are used in classical treatment in hospital. Another study talked about the molecular characterization of Enterobacteriaceae producing beta-lactamase and methicillin-resistant staphylococcus isolated from the hospital environment and specifically on catheter in some public hospital in Benin. So we bring out the invasive action of healthcare personnel introducing BMR via venous catheter. Another study talked about the surveillance and genomic of toxigenic Vibrio cholerae 01 from fish. And as you can see also, it's important to let people know that when they want to eat fishery product, they need to cook it well to maybe be sure at 40% that everything as bacteria has been destroyed before they consume it. So now, when we talk about aquatic environment, we are now, today we can say that antibiotic resistance and the chemical residues in aquatic environment in Africa and especially in Benin are polluted. We have a lot of aquatic systems that are polluted, but the main issue is how to remove these pollutants, if they are chemical and bacteriological, to reduce the burden of infectious disease in Africa. We found also a lot of resistance and virulence gene from water in southern Benin. And now we are working on equipment that can remove not only bacteria, but also can remove chemicals as antibiotic residues from this water. This is an experimental protocol that we are putting in place, and at the end we will see if it can be implemented. At hospital level, in the environment, we have the problem of effluent treatments, because we only have treatment system in maybe 2 or 3 hospitals in the country. So we are working on this to make the decision makers know that this is really important to avoid transmission from hospital to environment and now to human in the environment. So we have a lot of infectious risk and antimicrobial resistance in the environment, especially the area where we have hospitals. And at animal level, we were able to, at poultry and at poultry level, we were able to find some resistant genes, and especially zoonotic disease. We were able to give information about this infectious disease in poultry, and we also proposed some actions to take to avoid the transmission from animals to humans. And now at human level, we were able to sequence and analyze genes mediating extended spectrum by beta-lactamase, especially BLATM, SHV, and CTX-M production in isolate of antibacteriaceae in southern Benin. So let's talk now about the limitation we have in conducting AMR research. First of all, I want to say that I can say that we are the first unit now in Benin doing a lot to really fight against AMR with a local solution, but it's not always easy because we don't use sequencing and molecular typing techniques because we don't have the platform. For that, we need to go to another country, what is not always easy. We have a lack of formal proof of flow and transmission link between bacteria found maybe in hospital, in veterinarian sector, and also at human health level. So we have also the low involvement of human study because it's not really easy to conduct studies who involve human beings. The studies are limited to 1 or 2 components due to the high cost of such studies, relatively long time for such studies, and poor coordination of studies in the 3 sectors simultaneously. It's not easy to find people that are open-minded to work maybe with the lab technician, with the pharmacist, or with the veterinarian at the same time. It's not really easy. So our involvement in the fighting AMR in the 3 sector of One Health. Today, we can say that we were able to map the pollution of the main water courses by hospital effluent in the whole country from the north to the south. And also we have some projects, one that help us do some activities where we have, for example, the Erasmus Plus, who is a cooperation for innovation and the exchange of good practice. And this project aim to improve the quality of higher education in Benin and Burkina Faso to fight antimicrobial resistance by strengthening the human and technical capacities of the partner university. And then this project is being conducted now, and we have some professional and also some students involved. And also the laboratory testing, who was a real success. And this project aimed to strengthen the capacity of Beninese laboratories in antimicrobial resistant detection and blood testing. This was a real success. And this is some results. We were able to train biomedical laboratory technician in antibiotic sensitive testing and good practice in hospital. And also they get the ability to know how to use an antibiotic result to give good to give the medical doctor some tools to choose the right antibiotic for an infection. And also we used to do the awareness campaign of sepsis, on sepsis and antimicrobial resistance. And this is each year we choose a locality different from the one we used the last year, each year. And it's a real success also because it's like we are in the environment and we are trying to push, we are trying to give information, and we are helping people, especially health professionals, to get information. We have also a new project that's name is ICAS, who will help reduce the use of antibiotic in poultry farm in Benin in the way to reduce the impact of zoonotic disease. Now let me talk about the prospects in the fight against AMR. As prospects in research, we will do some study of the dissemination flow of carbapenem-resistant bacteria in hospital and community setting, and also study of the dissemination of Mycobacterium bovis between human and livestock and the environment in the north of the country. And also we will work on the genetic diversity of Salmonella strain circulating between human, livestock, and the environment. This will help us make the map of all the Salmonella strain we have in the country, and then we will see also the susceptibility to antibiotic, and then it will help us take the good decision to stop the spread of Salmonella. Because with this kind of bacteria, the zoonotics one are not really considered at the hospital level, and it's the opportunity for us to bring more light on this kind of bacteria. We also have the implementation of the MIME project that we train and that we intervene and raise awareness for the reduction of maternal and fetal mortality. And I already talked about the ICAS project that will be a joint work between veterinarian and health professional and researcher at the level of the university. So I want to say that we are really open to collaboration. We are open to host students for their work. And if you want to contact us, you can do it through our different channels. And also I will put in the chat our website and also the email address to help everyone access our information and contact us. So thank you for your attention. And I will take the question. I will try to answer them one by one. Thank you.
Okay, thank you, Dr. Adegeno. Thank you very much for this very interesting talk. I don't see the question in the chat. Okay. I don't see any question for the moment. Is there any question regarding the talk from Dr. Adegeno? Everything was so clear that we don't have any question, or maybe Fion, are you there?
Yes, I'm here. Yeah, there's no questions in the Q&A at the moment. I'm not sure if some people are having problems seeing the Q&A box, so feel free to put questions in the chat box as well if you're not able to use the Q&A.
Nice.
Thank you.
So, okay. I don't see any questions. So I think we will thank Dr. Deguenon again for the talk and being available for this trip, this roadshow. Now I will introduce the next scientific talk and invited speaker, Dr. Gerald Agbankpe from the University of Abomey-Calavi. He will be talking about phages biocontrol and therapy in a perspective of effective and sustainable control of bacterial infection and AMR in Africa. So Gerald will have to talk around 12 minutes, and we will have 3 minutes for the question. So Dr. Gerald, if you are online, you can share your screen and go ahead.
Okay, thank you, Professor Victorien Douillon. Let me share my screen. Can you see my screen?
Yes, perfectly. Now you can make it bigger. Okay, very good. Okay. You have the floor.
Okay. Thank you, everyone. I'm Gerald Agbankpe. I'm microbiologist. And I got my MIC in microbiology and food technology in 2012, and my PhD in microbiology and biotechnology in 2016. So I have more than 10 years experience in microbiology. Since 4 months ago, I'm head of phage and cell culture lab in my home research unit, which is a research unit in applied microbiology and pharmacology of natural substances in University of Abomey-Calavi. Then my research focus on foodborne infections, zoonosis, antimicrobial resistance, probiotics, and phage biocontrol. Currently, I'm African Research Excellence Fund Fellow and also ISOLOMSO Fellow. During my presentation, I will address 5 main points, starting with the problem of antimicrobial resistance and proposal for alternative solution to control AMR. Then I will discuss about phage and phage therapy with a focus on the case of Benin. And I will end my presentation with our capacity building in phage research. So any person at any age and in any countries can today be exposed to the risk of an ineffective traditional treatment against a bacterial infection, even a trivial one. A phenomenon that is accelerating both in humans and animals, simply because antimicrobial resistance is a natural phenomenon, but also because of the misuse of drugs. A risk that is also growing with the increase in the number of infection and the increasing and recurrent difficulty to treat them, which lead to longer hospitalizations, higher medical expenses, and increased mortality. According to ONEL's 2016 report, it is estimated that more than 2 million women illnesses per year are caused by antimicrobial resistance infection that result in approximately 700,000 deaths per year. If IMA continues to spray and check it by 2050, approximately 10 million people will die from antimicrobial resistance infection each year. This means that the number of deaths from antimicrobial resistance is expected to surpass cancer in a few decades if nothing is done. And as you can see, African and Asian countries will be the most affected because they are the countries where the hygiene and health infrastructures are insufficient or absent, or there is no clear regulation in terms of use of consumption of antimicrobial. So the population indulges in self-medication and antimicrobial misuse in both human and animal health. So to face this worrying problem of public health in the world, and especially in development country, the poor population since decades have done it to the use of medical plants to look after themselves at lower cost. Those scientists in the search for new antimicrobial have turned to medicinal plants, but since antimicrobial resistance is a phenomenon that results from random mutation and natural selection, the discovery of new antimicrobial will only buy us time. So antimicrobial resistance will not be slowed unless we develop alternative solutions to eliminate pathogenic bacteria carrying resistance gene. So one solution is the use of phages or phage therapy. So phage are virus which specially target and kill specific bacteria. Phage are easy to isolate and develop compared to antibiotic and reproduce at infection sites to ensure continuous dose supply of effective particles while being harmless to human and animal cells. Phage therapy was first implemented by Philips Derrel in 1917 to treat infection caused by Shigella dysentery. Phage therapy is the use of lytic phage to treat infection disease of bacterial origin, widely used as a miracle treatment in the 1920s to 1930s. It was gradually abandoned due to the discovery of antibiotics. So it was re-emergence in recent years as a future strategy in the face of resurgence of antibiotic resistant bacteria. So phage therapy is attractive as it has been shown to be effective at controlling multi-drug resistance human, animal, and plant pathogenic infection in many infection models. So what is the phage research in Benin? With our Pakistani partners, we had initiated the project entitled Potential Severity of Antimicrobial Resistance in the context of COVID-19 pandemic and phage-associated biocontrol, One Health approach in Benin and Pakistan, which was funded by the World Academy of Science and Islamic Development Bank in 2022. With this funding, we acquire equipment and consumables for the implementation of the project. Because that the equipment, what we need to do, the phage research is expensive equipment. So this funding allow us to implement this, to acquire this equipment. In addition, thanks to the leadership and support of our supervisor, Professor Victorien Douillon, we had established the first phage research lab in Benin in last November 2022. So as we can see, we have some picture of the laboratory with the equipment and the consumables. So after the lab was set up, we proceeded with the sampling for the isolation and phage. Phage are found in environments where bacteria grow. Those we sample community wastewater, animal farm wastewater, animal dropping, human feces, hospital effluents, etc. So after sampling, we centrifuge the samples and enrich the supernatant by contacting them with the specific bacteria in the liquid medium that we incubate at 37 degree under agitation for 24 to 48 hours. Then the mixture is centrifuged and supernatant is filtered and put in contact with the bacteria allow using the double layer overlay plate techniques. So the plates obtained after 18 hours of incubation are suspended in a pot, and the test is repeated 3 to 4 times for purification. So as you can see, we are able to isolate and purify lytic phage against multi-drug resistant strain of Pseudomonas aeruginosa, Klebsiella pneumoniae, Enterobacter cloacae, Acinetobacter baumannii, isolate in clinic. In addition, we also isolate lytic phage against multi-drug resistant Salmonella and Campylobacter strain isolate from animals. So the work is currently continuing on the phage isolates for the characterization. The objective of the project is to develop phage cocktail and test the efficacy in vivo in infected animal models. So in Africa, research in phages still started. So in terms of phage research in Benin, we need to strengthen our capacity for more in-depth research and collaborate with more experienced researchers in the field. So through the African Research Excellence Fund Fellowship, we benefit from a 9-month research stay at the University of Leicester in UK to first strengthen our skill in phage research, and secondly, to sequence our phage isolates and conduct more in-depth tests on them. And at the end of our stay, we will come back to Benin to implement this in deeply test and also building or strengthening the capacity of our students in our research unit. So moreover, this day will allow us to develop the partnership between the University of Leicester and the University of Abomey-Calavi. So I would like to end this presentation by thanking the Society Microbiology for giving me this opportunity to communicate of my research activities. Thank to my home institution, the University of Abomey-Calavi, and my research unit in applied microbiology and pharmacology of natural systems for the institutional support. Many thanks to the World Academy of Sciences and the Islamic Development Bank for funding this project. And my thanking also go to African Research Excellence Fund and also Stellenbosch Institute for Advanced Study for the support of my career development. And finally, I would like to thank the University of Leicester for hosting me for capacity building in phage research. So thank you for your kind attention.
Thank you, Dr. Gerald. We have a question. No, I think this one, Senam, was for Dr. Degeno. So Fion said that you should contact Dr. Degeno and go through the discussion together. So for Dr. Agbankpe, I don't see any question in the chat. We are going slowly in the road show. So any question for this presentation, or we can assume that everything was so good. One question: is great to see that Benin is picking up greatly on phage research. Which infection, Dr. Gerald, which infection do you consider as priority for your phage work? And how soon do you think phage therapy will be widely available?
Currently, we are focused on foodborne infections, especially Salmonellas infection and Campylobacter infection. So our plan is to start firstly about the phage biocontrol in animal health. And after that, if our result is interesting, we will go in slowly about the human health, because like we know, the human health is maybe very important. So we are going slowly for human health. So I can tell you when we will start the phage therapy on women. So we are trying to do slowly and go step by step. Thank you.
Okay, thank you. Sarah Cooper. Okay, I think there is no other question. So we will continue our exciting roads with the next speaker, Dr. Karambega from Burkina Faso. She's a microbiologist at the University Joseph Kizerbo. So, Dr. Aseta, if you are there, you can start sharing your PowerPoint. Dr. Karambega, are you here?
She's definitely in the room now. There we go.
Okay. So Dr. Aseta will talk about the genome analysis of Salmonella enteritidis, isolated from poultry and human in Burkina Faso. During 12 minutes, the same exercise, followed by a 3-minute question and answer. Dr. Aseta, if you are ready, then you have the floor.
Thank you, Dr. Douillon, for giving me the floor. I'm Dr. Aseta Karambega from the University of Ouagadougou, and from the food microbiology lab. I'm working on the epidemiology and antibiotic resistance of foodborne pathogens such as Salmonella, E. coli, Campylobacter, and Staphylococcus. And today I am going to speak about one study conducted in Burkina and in collaboration with the US Department of Agriculture. From this study, I have some little introduction about Salmonella. And I can say that diarrheal disease is a serious health problem that causes high rate of morbidity and mortality in developing countries. These diseases are due to the lack of hygiene and sanitation and have serious social and economic consequences that affect both individual and collective development. Salmonella is the main pathogen implicated in the diarrheal disease worldwide. Salmonella enteritidis is a zoonotic bacterium which can colonize human or animal through contaminated food, water, or direct contact with infected human or animal, especially poultry. Multi-drug resistant Salmonella enteritidis is of great importance in public health due to the resistance to several antimicrobial classes, which make treatment difficult. In Burkina Faso, multi-drug resistant Salmonella is responsible for death among children under 5 years old and immunocompromised people, but the data on the epidemiology of the strain is still very scarce. And however, the source and transmission route of Salmonella in developing countries are poorly understood due to the lack of coordinated national epidemiological surveillance system. Backyard and semi-intensive poultry rearing is gaining popularity in Burkina Faso, but little attention has been paid to the potential negative impact of zoonotic pathogen associated with poultry that can be transmitted to human through poultry droppings. The aim of this study was to investigate the genome of multi-drug resistant Salmonella enteritidis isolated from patient and poultry feces in Burkina Faso using the whole genome sequencing. The bacterial strain was isolated from poultry and patient using standard method, and serotyping was done after whole genome sequencing using some 6-0 as described by Karambega et al. 2021. And DNA extraction for sequencing genome sequencing, assembly, and annotation. The genomic DNA was isolated using the GeneLith bacterial genomic DNA kits, and the Illumina library were prepared using the NextTera kit. And identification of antibiotic resistance, virulence, chromosomal mutation, serotyping, MLST, and plasmid from total genome sequence was done using the responder, the 60, and also the plasmid finder. The genome sequence were analyzed for potential impact profiles using the web-based profiles predicting tools, the FASTER. Like a result, the Salmonella enteritidis isolate are both gene conferring resistance to aminoglycosides, chloramphenicol, trimethoprim, tetracycline, streptomycin, and also some known mutation in different candidate genes imparting resistance to different antibiotics were found in all strains. More than 25 different virulence factor genes were identified in each strain. And the replicon type were identified in all the strains. Two types of replicon, of plasmid replicon like the ink. In total, 4 types of profiles were identified in each strain also. And in discussion, the sequencing data show that Salmonella enteritidis sequence type 11 isolate can be obtained from chicken feces slaughtered at the common marketplace in Ouagadougou, as well as from the feces of diarrhea local patients. These facts show the length of cross-contamination between poultry and human, trout, consumption of poultry product, and or by direct contact. The strains show multi-drug resistance. The strains show multi-drug resistance, and this could be explained by the uncontrolled use of antibiotic for poultry farm and human. And here is some picture from marketplace during poultry carcass processing. Thank you for your attention.
Thank you very much, Dr. Aseta. Any question? Looking for the chat. Don't know if we have any question. Okay, that means that everything was so clear. We don't have any question. Okay, I would like to thank Prof. Okeke. Well, your question is not complete. We see a lot of enteritidis ST11 infection from blood. Do you please?
I was just asking, thank you for the talk. I was just asking whether you see a lot of systematic blood infections of Salmonella enteritidis ST11. Thank you.
Okay, thank you very much. Dr. Aseta?
Yeah, yes.
Do you get the question?
No.
Do you have this situation?
What situation?
A lot of enteritidis ST11 infection from blood.
Yes, for the children under 5 years old, it's very common.
Okay, and another question is about what is the relevance of the different AMR genes observed from the whole generational sequencing? Genome sequencing.
Yeah, the resistance gene we found, we first done the phenotypical method, and we take only the resistant bacteria to do the whole genome sequencing, and using the responder, we found again the resistance gene, confirming the phenotypic resistance found previously.
So those genes are more prevalent in Burkina Faso, or on which?
Yes, yes,
basis
it's some the antibiotic, they are so common in Burkina Faso, and I think that's why we are using to test our strain also, because they are antibiotic commonly used for animals and for human also. They are the same.
Okay. Thank you very much. I think at this step, I will take still one question. How does your data compare with previous related study? Any other strains?
Yeah, the data compared to other related studies, we can say that related studies find also like similar results for some of our study. We found that like poultry, salmonella isolated from poultry, they are similar. Like the genetic relatedness is very high from salmonella isolated from poultry and human. And that is similar to other study also. And other strain, we have some other strain like Campylobacter, but Campylobacter also we found out that they are high. We find high prevalence from poultry, but not for other animals. We only done Campylobacter from poultry.
Okay. Thank you very much. If there is no other question, I'm very pleased to thank all the speakers and the attendees for their question. Now we will move to the final section of the day. So I'm happy to check for Philip and Omolulu.
Yes, I am there.
Okay. And Dr. Omolulu?
Yes, I'm here.
Okay, so I think it was a wonderful session. I would like to give you the opportunity to say some word before I give the floor to Peter for the last word of this session. Thank you. Philip?
Okay, thank you so much, Professor Bouillon. I would like to thank all participants for attending these meetings. I'm also very grateful to the Microbiology Society for the opportunity given to us for organize these events. I'm also grateful to Fifion, who has done great for this event. I'm very glad to be here, and I hope we will do great for the promotion of the Microbiology Society. Thank you so much.
Thank you. Dr. Omolulu?
Yeah, I really want to thank, it's such a successful event today. I'm really excited that we all made this to be possible, especially for those people that are hearing about Microbiology Society for the first time, or they are attending Microbiology Society. I want to say a very big thank you. for trusting the society and what the society has to offer. I believe that you must have benefited from the various talks in this program. Thank you to all our speakers. Special thank you to the CEO of Microbiology Society. And if I look at a common language in West Africa, that would be Hausa, possibly, especially in the northern aspect of West Africa. So on that note, I would say Segobe. Good night or bye-bye.
Segobe, thank you very much. And it's my turn to thank everyone here in this session, from Fionn, who was really hard pushing and believing in this activity. I think it was a great success, and thank you all the speakers, the participants. I will leave the floor to Peter for his words, and see you next time for another activity. Thank you.
Thank you very much indeed, Victorian. I have had a fantastic day. It has been absolutely fascinating. I have learned all sorts of things, and I've really enjoyed meeting people. We always want to make our events at the Microbiology Society the best they can possibly be. And one of the ways we do that is we ask for feedback. And so there will be an evaluation survey, which will, I think, will appear in the chat right now. If Fionn, yeah, there we are. Fionn's done, look at that. Fionn has done it right away, but we'll also email it around after. If you can't get it now, we'll email it around afterwards. But do please give us some feedback, because we always want to make our events better and better every time. I want to reiterate what's just been said about thanking people. I want to thank Fionn and all the staff for doing a fantastic job. I want to thank Philippe and Victorian and Omolulu for everything that they have done. I want to thank all of our fantastic speakers, Del, Iruka, Gale, Marguerite, Adebare, Michael, Joseph, Esther, Gerald, and Aseta, all of whom have given us really interesting things to think about. So thank you all very much indeed, and have a good weekend. And I look forward to seeing you at the next Microbiology Society event.
Thank you so much.